Hello, this is Dr. Deborah Morris, and today we're going to be talking about the physiology and the anatomy of the nervous system. So, because this is a pretty complex presentation, I'm dividing it into two parts, but the two parts comprise what is lecture 4 on the syllabus. So, we'll start by focusing on neurons, the action potential, the synapse, and neurotransmitters. And then we'll move on to talk about the embryology of the central and peripheral nervous systems, primarily the central nervous system, and the gross structure of both the central and peripheral nervous systems, and then the sensory motor system and the motor pathways, and how that information is transmitted within the central and peripheral nervous systems. I like to start this lecture with this drawing because it was made in the 19th century. Somebody using a microscope and examining uh, layers of the brain drew pictures of neurons, and you can see from the detail in these pictures that there are some different conformations, different shapes to these neurons. Some of them are quite stellate, and some of them have this triangular cell body. They all have extensions, the extensions being axons and dendrites. And even though at that time very little was known about the function of the nervous system, it was apparent to anybody doing this kind of work that the nervous system was extremely complex. And in fact, it is the nervous system of a human being the most complex system known to man. Neurons have parts, and they are a little different from many of the other cells that you've studied. Certainly, they have a soma, or a cell body, within which is not only a nucleus with the genetic material, but also all the other organelles you might expect in a highly functional metabolic cell. So there are lots of mitochondria because neurons use a ton of energy, so they need a lot of little um, energy machines. And then there is all of the cellular apparatus that that allows synthesis. So you have endoplasmic reticulum, you have lots of ribosomes, and you have Golgi apparatus, and you have all kinds of RNA tooling around in here, RNA in the nucleus translating the DNA, and then RNA transmitting messages, carrying amino acids, and assembling amino acids, and other substances get assembled and processed in the endoplasmic reticulum and the Golgi apparatus. And then actually neurotransmitters that are created in the soma of the nucleus end up getting transmitted along the axon to the axon terminals. You notice these many structures, and most neurons have multiple dendrites. The word dendrite is derived from Greek for tree, and you can see that they have the appearance of a leafless, a winter tree, and that there are multiple dendrites on the neuron. However, there is only one axon. Dendrites receive messages, and by and large, axons transmit messages. So the action potential that is generated in the neurolemma or cell membrane of the cell body is transmitted along an axon. This particular axon is myelinated, meaning it has fatty cells wrapped around the axon fiber with small spaces between them. Those cells you can see have their own nuclei, and they create something called the myelin sheath. In the peripheral nervous system, The cells that make up the myelin sheath are Schwann cells. The spaces between the Schwann cells making up the heavy myelin along this axon are called nodes of Ranvier, and they allow for something called saltatory conduction that speeds up the electrical transmission of signals along the axon. The axon terminals are where neurotransmitters are released into the synapse. Axon terminals may synapse with cell bodies. They may synapse with dendrites. Dendrites sometimes synapse with other dendrites. And there are different conformations of connections. But the primary model is that axons are the transmitters, that they synapse with either muscles, releasing acetylcholine at the neuromuscular junction, or with, and that's in the skeletal or voluntary muscle system, or they terminate at various places on dendrites or on cell bodies. Again, this is a similar slide showing you the cell body, the soma, containing all of the necessary organelles, the dendrites, which are the primary receivers, and the axon, which is a single extension of the neuron, which may or may not be myelinated. Some axons are bare, some axons have light myelin, and some axons have heavy myelin. There is a structure in the cell body called the axon hillock, which is the beginning of the extension from which the axon extends. Just as the dendrites tend to be heavily branched, the terminal axon branches. Now, there are different shapes of neurons. I'm going to flip back here so that we can see that very first slide. And you can see these different shapes of neurons, but different kind of conformations. Some of them have tons and tons of dendrites, some of them have fewer dendrites. We call them by somewhat different names, and the names are not terribly important, but their function is terribly important. So I'm going to, for the purposes of this lecture, define three types of neurons. Okay. A motor neuron is a unipolar neuron. It has a cell body, it has dendrites, it has a single long axon that terminates at the neuromuscular junction, at the synapse between the motor neuron and skeletal muscle. Same is true in the autonomic nervous system, terminating at smooth muscle in many cases. There are sensory neurons. Motor neurons are also considered effectors. They are messages out. So in this case, from the central nervous system to perhaps the peripheral nervous system, a motor neuron. There are lower motor neurons in the peripheral nervous system and upper motor neurons in the central nervous system. The lower motor neuron terminates at the muscle. Sensory neurons are somewhat different. They are effectors. Their signal begins at a receptor cell. That receptor cell might be in the skin, it might be in the eyeball, it might be in the cochlea, but the receptor cell then sends signals down the axon of the sensory neuron in what is called a pseudo-unipolar neuron. In a pseudo-unipolar sensory neuron, the cell body is in the center, and the axon goes from the receptors into the central nervous system. So starting generally in the peripheral nervous system and ending in the central nervous system, with a cell body that is sitting in a group of cell bodies near the nerve root, outside of the spinal cord, outside of the central nervous system. The other kinds of neurons, besides motor neurons and sensory neurons, are interneurons. And interneurons are called multipolar. They again have a single axon, though that axon may be very branched with multiple axon terminals. They have different dendrites, or lots of dendrites, but the dendrites may be configured differently. And these neurons exist between other neurons, so they're interneurons. In the example of the deep tendon reflexes that we all laugh about, when somebody taps your knee, it stretches a tendon, triggering stretch receptors, which then send a message to the spinal cord, with the cell body sitting in the dorsal root ganglion, and into the spinal cord, where the axon terminals synapse with an interneuron, which then carries the message to a motor neuron, which sends its axon out back to the muscle, causing it to contract. So there are 3 neurons involved in that reflex loop: a sensory neuron, an interneuron, and a motor neuron. In the central nervous system, there are lots and lots of interneurons, with also some motor neurons and some other neurons that are collecting information and processing information. In all cases, neurons communicate with each other through electrical and chemical messages. They send an action potential down the axon that releases a chemical into the synapse that either activates, starts a new action potential, or inhibits and stops the action potential at the next neuron. Different neurotransmitters depending on receptors, as we talked about in the ANS, may be inhibitory or stimulatory. The synapse is the tiny gap between the terminal axon and whatever it's communicating with, whether it is a dendrite, a cell body, or a muscle cell. And so here we are showing a neuron that we're calling, in this case, the presynaptic neuron, with this next neuron being the postsynaptic neuron. Something has happened to trigger an action potential at the axon hillock, which is going to send that message electrically down the axon to the places where the terminal axons synapse with the next cell, the postsynaptic neuron. When we were talking about the autonomic nervous system, the presynaptic neuron might be in the spinal cord, in one of the nuclei for at say the level T1. The postsynaptic neuron in that case would be in the sympathetic chain ganglia. So we have a presynaptic neuron sending its message to the postsynaptic neuron, where the terminal axons interact with little extensions that are either on the dendrites or sometimes even on the soma, that are called dendritic spines. So dendrites have little bumps on them called dendritic spines, which is where the axon terminals join and create a synapse. And a synapse is a space. So they don't touch each other. There's always a space, and that space is filled with extracellular fluid. Notice in this diagram, this is a colorized electron micrograph. This is a diagram showing the terminal axon in the presynaptic cell. The terminal can also be called a bouton. There are mitochondria in the terminal because there is energy being used. There are vesicles, and those vesicles contain neurotransmitters that was made up here in the cell body and transported along microtubules down the length of the axon to the presynaptic terminal. When the action potential reaches here, it opens gated calcium channels, which I'll show you in a minute, that cause the synaptic vesicles to fuse with the neurolemma, with the cell membrane. And that allows these chemical messengers to be released into the synapse. So in this micrograph, you can see that space, the synaptic cleft, you can see the vesicles, you can see a mitochondria. You don't particularly see the vesicles merging with the cell membrane. Maybe we have one there and there. In the postsynaptic membrane, there are receptors. The neurotransmitter molecules bind with the receptors, and then something happens here. Either a new action potential is created by those chemical messengers opening chemically gated or ligand gated channels to allow an influx of sodium to start a new action potential that then travels to the cell body along the dendrite. The next couple slides are similar to what we talked about in the autonomic nervous system. They're a little less detailed. So you remember that all neurons, in fact all cells in the body, have sodium potassium ATPases that are constantly pumping out 3 sodium ions for every 2 sodium ions that get pumped in. That creates a negative charge within the cell, and that negative charge often sort of sticks to proteins. So there are negatively charged proteins in the cytoplasm. And I remember mentioning when we were talking about the composition of intracellular and extracellular fluid, remarking that there is a lot more protein in the intracellular fluid than in the extracellular fluid, and those protein molecules are negatively charged. There are channels in the cell membrane, and many of those channels are potassium channels, and because of the sodium potassium ATPase, we have a constant movement of potassium that was pumped into the cell back out of the cell down its concentration gradient from high potassium inside the cell through the channels back out of the cell, which is removing more positive charges. There are also sodium channels, but those sodium channels are usually closed. Some of them are ligand chemically gated, some of them are voltage gated. When the potassium and sodium ions and the chemical gradient and the electrical gradient are in balance, we reach something called the equilibrium potential. And so you have some potassium flowing out of the cell down its concentration gradient. You have some potassium flowing into the cell being attracted by negative proteins, but you reach an equilibrium where the same amount of potassium is leaving as is coming in. And that establishes this charge, which for potassium is actually minus 90 millivolts, but when we consider the potassium and the sodium channels and everything that's going on, in most cells it's about minus 60 millivolts. So there's a charge, negative charge on the inside of the cell. Just as the negative charge on the inside of the cell is attracting potassium ions, it's attracting sodium ions, but those sodium channels are closed. However, if a neurotransmitter, a chemical ligand binds with a ligand-gated sodium channel, that sodium starts rushing in. And it's rushing in for two reasons. One is it's going down its concentration gradient from high sodium outside the cell to low sodium inside the cell. And two, it's going down an electrical gradient from the positive charge on the outside of the cell to the negative charge on the inside of the cell. When the sodium comes into the cell, it causes depolarization. That negative charge actually reverses, goes to zero and into positive territory. If there is enough of a positive charge and you reach threshold, that's when the action potential occurs. You can have small numbers of neurotransmitters that don't open enough sodium channels to bring the charge to threshold. But once it's brought to threshold, then suddenly the voltage gated sodium channels start opening and an action potential begins to propagate down the cell membrane of the axon. The axons that are naked, that's just direct. The charge propagates along the cell wall from the beginning, from the axon hillock to the terminal. But in myelinated axons that have these segments that are highly insulated and then only have sodium channels at the nodes of Ranvier, the charge jumps. It kind of jumps from one node to the next node, and that creates a much faster transmission of the electrical signal along the axon. So motor neurons, for instance, the neurons that go from your spinal cord to your feet and make you move your big toe, they're long, they're heavily myelinated, but the heavy myelination allows for that signal to move extremely quickly. Not instantaneously, but extremely quickly. 7 meters per second here. Okay. At the terminal axon are the vesicles that contain neurotransmitters. As I mentioned earlier, the action potential that reaches the terminal axon opens voltage-gated calcium channels, and the calcium is what works with the vesicles to make them merge with the cell membrane, releasing the neurotransmitter into the synaptic cleft where it's going to interact with receptors on the postsynaptic cell. That cleft is really tiny. I had this slide saying that the synaptic cleft is 20 nanometers wide, so I decided to look up how wide is a human hair. And a human hair is about 75,000 nanometers. So this is a very tiny infinitesimal space containing extracellular fluid. As I mentioned earlier, the human nervous system is the most complex system known to man, and we don't fully understand it. But each neuron synapses with hundreds to thousands of other neurons in the central nervous system. And this creates something that has become known as neural nets or networks. And it's estimated that the human brain has between 100 and 1,000 trillion synapses, and those synapses are constantly being edited, rewired. Every experience you have, every memory that you form results in some change in neural nets in synapses to allow the experience to go into memory. So there's constant change in these neural nets. And this is just everything that we've talked about. One of the things that you can see here that we didn't see in some of the other diagrams are these filaments that are protein filaments, microfilaments that transport the synaptic vesicles from the cell body down to the axon terminal. So we have the synaptic vesicles being transported to the synaptic terminal. We have an action potential reaching the terminal. It opens electrically gated calcium channels, which allow calcium into the cell. Remember that there's a lot of calcium outside the cell and not much at all, very, very little in the intracellular fluid. That causes the synaptic vesicles to merge with the cell membrane, kind of turning inside out and releasing the neurotransmitters into the synaptic cleft where they interact with receptors. Those receptors might cause a new action potential by stimulating. They might cause um, the signal to get stopped by inhibiting. Um, so different things can happen, but the neurotransmitter then has to get removed from the synaptic cleft, and it may be removed by things like we talked about acetylcholinesterase, which destroys the neurotransmitters. It might be retaken up by the axon terminal and repackaged in vesicles to be reused. This slide just illustrates some of the major neurotransmitters that are used in the nervous system, both the central and peripheral nervous systems. We've already talked about acetylcholine, which is an important neurotransmitter in several systems. We know about its use in the autonomic nervous system, both in the sympathetic and parasympathetic arms, at the presynaptic neuron for both arms, and at most of the postsynaptic, if not all, in the parasympathetic arm, and at sweat glands on the sympathetic side, and at every skeletal muscle neuromuscular junction. It is in most of its places excitatory, but depending upon the site and the neurotransmitter, there are places where it might be inhibitory. There are a group of neurotransmitters we call biogenic amines. One of them is norepinephrine, and you remember I also called norepinephrine and epinephrine and their relatives catecholamines. They are amines. They have an amine group with side chains, hydroxyl groups, and there are several of them. There are 3 illustrated here, including norepinephrine, again, excitatory or inhibitory depending on the site and the receptor. Dopamine, which at most sites is excitatory, is extremely important in movement. So the neurons of the substantia nigra, which get destroyed in Parkinson's disease, which is at least partly a movement disorder, are dopaminergic neurons. It's very important for mood, and many psychiatric medications are agonists or antagonists of dopamine. And then serotonin, which is very involved in a number of things, including mood, which is why, again, there are drugs that are serotonin agonists and serotonin antagonists. There are serotonin reuptake inhibitors, which function as agonists by increasing the amount of serotonin in synaptic clefts in relevant places. So there are tons of drugs that interact with all of these biogenic amines as either agonists or antagonists. There are a number of amino acids that function as neurotransmitters. So again, they get either synthesized or collected in the cell body, transported down the axon, stored in synaptic vesicles. These include GABA, which is gamma aminobutyric acid. It's an inhibitory neurotransmitter. Again, there are a number of drugs, psychiatric drugs, including the benzodiazepines, which interact with GABA receptors, may be agonists or antagonists depending on the drug. Other amino acids which function as neurotransmitters include glycine, glutamate, and aspartate. And then there are the neuropeptides. There are a ton of neuropeptides. So these are short chains of amino acids. They're not proteins, but they are short chains of amino acids that function as neurotransmitters. And among those that we will find ourselves hearing about or talking about frequently are two that are very involved in pain perception. Substance P, which is important in generating and transmitting pain signals, and enkephalin, which is an endorphin or endogenous morphine like substance, which actually inhibits the perception of pain signals. So, those are just two examples. I'm going to end here and record the next part of this lecture where we talk about neuroanatomy.